is Amprasad Naidu. Today we are discussing about the cytokine that roles in rheumatoid arthritis and inflammatory disease, and cytokines role in separate health disease. Cytokines are small secreted proteins which are mediated and regulate the immunity, inflammation, and hematopoiesis. They are produced in the response to an immune stimulus. Cytokines, in the general terms, as a large group of molecules involved in the signaling, is like a signaling between the cells during the immune response. All cytokines are proteins, some with sugar molecules attached with the glycoproteins. They are synthesized and released by white blood cells and tissues macrophages. They are proteins, peptides, glycoproteins in nature. Cytokines stimulate or suppress the functional activity lymphocytes and monocytes, neutrophils, fibroblasts, and endothelial cells. They modulate the functions of the other cell types, long and known as to be involved in the cellular immune response. These products have additional effects that play an important role in both acute and chronic inflammation. 200 different human cytokines have been identified. That means 200. So not only 1 and 2 or 3. 200 human cytokines have been identified. The terms and definition, these cytokines are generated by the mononuclear phagocytes and often called as chemokines, and those activated lymphokines are referred to as a lymphokines. In additionally, both monocyte and macrophage produce the cytochromes cytokines such as the CSF, which stimulate the growth of immature leukocytes in the bone marrow. So general properties of cytokines are produced during the inflammation, immune and inflammatory response, and secretion. These mediators, the transient, closely regulated. Cytokines effects are often reabundant. These proteins are influence the scale, synthesis or actions of other cytokines. Cytokines mediate their effects by the additive, binding to specific receptors or target cells, and the expression cytokines receptors can be regulated by the variety exogenous and endogenous signals. Cytokines include induce their effects on in two ways. They act on the same color the product autocrine effect, and interleukin 2 produced by activity of T cells promotes and T cells growth. They affect the effect of the other cells in their vecnative parakine effect, and interleukin 7 produced by the marrow stromal cells promotes the differentiation of beta cells and progenitor in the bone marrow. The many classic growth factors act as a cytokines. Conversely, many cytokines have growth promoting properties. The main steps of cytokines here is interleukins, interferons, tumor necrosis factors growth factor, colony stimulating factor, and chemokines. These are the major factors. Coming to the first one is interferons. The interferons or proteins are released by the helper CD4 cell T lymphocytes as well as through monocytes, macrophages, endothelial cells, and response to presence of pathogens such as a virus, bacteria, or parasites or tumor cells. Interferons belongs to a large class of glycoproteins known as cytokines, although they are named after their ability to interfere with viral replication within host cells. They are produced very early in infection, and they are fast to assistant great many viruses. These are particularly important limiting spread and certain viral infections. One group interferon and interferon alpha, interferon gamma produce cells which have become virally infected. The types of interferons, some different types of interferons. Based on the type of receptors, human interferon have been classified 2 major types, that is interferon type 1, interferon type 2. Interferon type 1 is all types of interferon binds with the specific cells of a receptors complex known as interferon alpha. Alpha type 1 is interferons present in the human, interferon alpha, beta interferon gamma. So here mu. So omega. Interferon type 2 is only human. This is interferon gamma. interferon 1 is the alpha, beta and omega, and the interferon interferon type 2 is only gamma. The functions of interferon activate the immune cells such as the natural killer cells and macrophages. They increase the recognizes of infections of tumor cells and upregulations of antigen presenting of type 2 lymphocytes, and also they increase the ability of uninfected hosts host cells resistance new infections of by viruses. Certain host systems such as achieving muscular and fever and related to the productions of interferon during the infections. So these are the coming to the interferons. So the interferon type 1 and type 2. Coming to the interleukins here, these are the large group of cytokines produced mainly T cells, although some of the also produce mononuclear phagocytes by tissue cells. The interleukins were first described as a signals communication between the white blood cells and leucoforms and leukocytes. Currently, it was well known that data molecules produced used as a signaling molecules in many cells of the body. In addition, immune cells they have. Finally, we're talking about so some of the interleukins. So they have a variety of functions, but most of them involved in the directly their cells is differentiate. Interleukins represent a broad family of cytokines that are made up of hemopoietic cells and act as primarily on leukocytes. There are currently 35 well-known interleukin cells. However, there are many more to be found in characterized. The tumor coming to the tumor necrosis factor. Tumor necrosis factor is a family of refers to group of cytokines a family of can be cause death. Tumor necrosis factors act as via the tumor necrosis factor receptors and part of the extensive pathway of trigger apoptosis. TNF a tumor necrosis factor interact with the tumor cells, trigger cytolysis or cell death. So types of tumor necrosis factor alpha, and the most well-known number of classes are sometimes referred to as when term as tumor necrosis factor is used. Tumor necrosis beta is also known as lymphotoxin and also cytokines that is including by the interleukins 10. Colony stimulating factors. So what is colony stimulating factor? It is secreted by the glycoproteins which bind the receptors proteins surface the hematopoietic stem cell and thereby activated intracellular signaling pathway which can be caused to proliferate and differentiate with the specific kind of blood cells. These are the primarily involving directly divisions from the differentiating with the bone marrow and stem cells and the previous precursors of blood cell leukocytes. So how means the colony stimulating factors are these are the precursors of bone marrow precursors of blood leukocytes. So types of CSF colony stimulating factor 1, 2 and 3. Colony stimulating factors of macrophages colony stimulating factors, and the second one is granules and macrophages stimulating with the also GMSF colony stimulating factors, and segregating and colony stimulating factors of colony it is called as filtration. So growth factor. What is growth factor? Growth factor is a naturally occurring substances capable of stimulating cell growth and proliferations of cellular differentiation. Growth factor typically act as signaling molecules, and between the cells they are important regulating variety cellular process. And epidermal growth factor, fibroblast growth factor, and granulocyte colony stimulating factor, and nerve growth factor. These are the growth factors. Epidermal growth factor, fibroblast growth factor, granulocyte a colony stimulating factor, and nerve growth factor. These are the growth factor. Coming to chemokines, this is the large group of chemotoxic cytokines directly movements of cells and around the blood body from the bloodstream into the tissues appropriate location with the each tissues. Some of the chemokines also activate the cells and carry out of the particularly functions. Chemokines are cytokines that are share ability to stimulate the leukocytes movements, and chemokines are directly movement of chemotoxins are particularly important in inflammation. Cytokines receptors type 1 and type 2, type 3, type 4, and these are the 4 types of cytokine receptors. The type 1 receptors are the cytokine receptors, and largely cytokine receptors and type 1 receptors superficially characterized by the extracellular region and structurally homology of approximately 200 amino acids long. Receptor cytokines interleukins 2, 3, 4 5, 6, 7 and 9 and 12 and granulocytes and colony stimulating factor, and granulocyte colony stimulating bag belongs to this family, type 1 family, and type 2 and type 3, and this type 2 is family related to the cytokine receptor can be considered as the part of immunoglobulin super family, and it contains the receptors of all interferon gamma types of, as well as interleukins 10 and colony stimulating factor. Type 3 is the responsible it receptors tumor necrosis factor cytokines binds with the type 3 cytokines receptor, which is all have multiple system like a cysteine which repeats the 40 amino acids in the extracellular domain. So type 4 is the receptors of interleukins 1 1 alpha and beta representative type 4 cytokine receptor family. Regardless these subtypes of cytokines receptors have several common characteristics. They usually consist 2 or more subunit receptors for different cytokines, may even the share common subunits. Some of the mechanisms involving cell activations, the binding of cytokine to its appropriate receptors of a cascade that leads to inductions and inhibition, transcription, a number of cytokines regulated genes. This occurs via the chain of promoting protein and protein recognizing events leading to binds with the diverse transcription factor DNA. Cytokines initiating the intracellular signaling through the ligand induced aggregation of receptors components. Cytokines binding with can cause the hetero and homogenic dimensions of receptors, and terminalization depends on a particular family. So how this is all regulated, some of the clinical aspects, coming to the clinical aspects of rheumatoid arthritis. Rheumatoid arthritis is a chronic systemic inflammatory disorders. It may be effect on tissues and organs, but principally attacks the joints, and an autoimmune reactions in the which T cells play pivotal role widely held to responsible for a chronic destructive nature of rheumatoid arthritis. Once an inflammatory synovites have been initiated by an exogenous agents. So T cell CD4 memory cells appears with the rheumatoid effects of giants, and early in the development of rheumatoid arthritis. Soon endothelial synovial giant capillary activated within the expression of ICAM it leads to attach the migrations of inflammatory cells that leads to sequence of further enhanced to the release of interleukins 1 and TNF alpha and interferon gamma. Activated the CD4 stimulated activate the monocytes and macrophages and promote that these release monokines and activated beta cells and antibody production in effects of giants. So tumor necrosis factor alpha and have been induced the resorptions of cartilage and bone, and by stimulating the releasing of collagenase and form the synovial cells, and upregulate the expressions of adhesions of molecules like ICAM1, which inhibits the synthesis of proteoglycans in the cartilage, stimulate the fibroblasts via the platelet derived growth factor. So multiple myeloma, it is also one of the cancer. Loma means cancer. So how it is malignant. See, multiple myeloma is a plasma cells cancer originates in the bone marrow and is characterized by involvement of the skeleton and multiple sites. So the proliferation, the differentiation of myeloma is cells depends of several cytokines, mostly notable in not only this interleukin 6 serum levels. These cytokines are increased in the patients which with active diseases. So interleukin 6 seems to be produced of tumors plasma cells themselves as well by the fibroblasts and the macrophages surrounding the stroma. In addition to causing the growth myeloma cells, and cytokines are also mediate the destructions by the acting of osteoblast and activating the factors tumor necrosis factor beta and interleukins 1 and interleukin 6 and macrophages colony stimulating factor. Some cytokine therapy here we are talking about cytokine therapy. Non-specific stimulations or inhibitory particularly components in the immune system may sometimes be benefits. The best results have been obtained with the cytokines among the interferon alpha, mostly widely mainly it is the antiviral property. The most striking clinical effect of cytokine has been that growth stimulating factor, colony stimulating factor, restoring bone marrow functions after the anti-cancer therapy. So interferon beta 1 interferon beta B also used the treatment of control of multiple sclerosis and autoimmune disorders, and interferon therapy is used from the combination therapy of chemotherapy, radiation, and as treatment of many cancers. These treatments most effective for the treatment of hematological malignancy, leukemia, and lymphomas, and including the hairy cell leukemia, chronic myeloid leukemia, and nodular lymphoma, and cutaneous T-cell lymphoma. Both hepatitis B and hepatitis C are treated with the interferon alpha, often in combination with the antiviral drugs. So some of administrated intravenously in low doses of interferon extensively in the Eastern Europe and Russia, methods to prevent the treatment of viral respiratory disease such as cold, flu. Interferon therapy causes about the immune suppressions, in particularly through the neutropenia, and can result in some of the infections of manifestations in usually discover some of the discoveries beta and rates HIV1 these are the conditions. So cytokines, some inhibitors, we were talking about the inhibitors can be used from the several chronic inflammatory conditions. Various inhibitors like TNF-alpha and interleukins 1 have been proved the variable rheumatoid arthritis, and more controversially is septic shock and also severe malaria, and cyclosporin and also pentamidine. So these are the conditions about the cytokines and classification, and cytochrome classification and cytochrome disorders, and how to treat and how to inhibit. These are the relations about the cytochrome.